All right, so this is where we get into specific GPCRs and what they do. So behind me are in the big cells are the dopaminergic neurons, and they secrete dopamine, and there are dopamine receptors. Dopamine receptors are all GPCRs. What exactly do they do? So let's go ahead and dive in. So just as a reminder, like we have all these things, all these receptors that we've mentioned or talked about before, and they're all metabotropic, right? So the muscarinic, there's like 5 different subtypes. The or muscarinic acetylcholine receptors, they're all metabotropic. mGluRs, so metabotropic. Glutamate receptors, there's like 8 kinds. The GABA B receptor, there's several kinds. Dopamine, serotonin, you can see how many kinds there are. And these are all going to do perhaps different things. So like, for example, the D1 and the D2 dopamine receptors have opposite effects. But and at this point you might be like, oh my gosh, like how many of these do I need to know? This is a lot. Well, I have good news for you because all of this can be boiled down into 3 types and with 3 different effects. So all of the receptors on the previous page either have use the GS, GQ, or GI G protein. So all of these receptors are different, but each of them will bind to only one of these 3 G proteins, and that is going to determine their effects on the cell. Okay, so GS, GQ, and GI, you can see here they have different effects, but one thing I want to point out, they all make use of something called second messengers. So cyclic AMP is a second messenger, calcium is the second messenger. What exactly is a second messenger? So second messengers are small, non-protein hydrophilic molecules or ions. Why is that important? They're small and they can go inside the cell, and so they can go diffuse pretty rapidly and activate proteins throughout a cell. So here's an example. And they're called second messengers because like the neurotransmitter is like the first messenger. So this is the second messenger, and it goes inside the cell to go activate things and have effects. And so, for example, a G protein, this G protein activates cyclic AMP. The cyclic AMP will go and activate various things inside the cell. Each enzyme can make a lot of second messengers. So a nice side effect is that you get amplification. And so just think about this for a moment. Can the activation of one GPCR molecule cause phosphorylation of dozens of proteins? Yes, it can. And so what happens is this process of amplification. So the receptor is on for a certain amount of time. It's going to activate not just one G protein, but like many G proteins, one after another. And then each G protein is going to go and act on its target. Okay. And then that target may make a lot of second messenger. Okay, that's what this blue to green is going on. And then each second messenger may go and activate many of its target, for example, a kinase. And so in this way, activation of one receptor once can cause, because it goes through several stages of amplification, it can cause activation of dozens or hundreds or maybe even 1,000 different target proteins, like individual proteins. And so each of those pathways there that I showed, the GS, the GI, and the GQ influences different second messengers. So as we'll see, GQ influences calcium, IP3, and diacylglycerol, while the GS and GO influence mostly cyclic AMP, but a little cyclic AMP. And this slide is just, if you're curious, some targets of each of these second messengers. So we'll go through the important ones or the key ones that are used in many pathways in more detail. So let's start with GS and GI. Sometimes GI is also called GIO. O stands for olfaction. Those are the ones used by the receptors in your nose. That's all. Um, so I grouped them together because they both work on cyclic AMP, but in opposite directions. So the GS activates a enzyme called adenylyl cyclase. Adenylyl cyclase makes cyclic AMP. And so the adenylyl cyclase will make cyclic AMP from ATP, like that. Cyclic AMP has a variety of effects in the cell. So it can open up certain channels, but it can also activate pKa, which is a certain phosphase. Sorry, a certain kinase, pKa. So protein kinase A, and pKa phosphorylates a lot of targets. The I is opposite. So what it does is it inhibits adenylyl cyclase. So all of this is opposite. So you have less cyclic AMP, you will have less activation of protein kinase A, and overall there would be less protein phosphorylation by protein kinase A. GIO also has other effects. So the effects I showed you here were all from the alpha subunit. So the DS, this is what the alpha subunit does, activates adenylyl cyclase. For GI, this is what the alpha subunit does, is it inhibits adenylyl cyclase. And so for GIO, actually the beta gamma complex also has effects. So here, GS, it activates GIO, inhibits adenylyl cyclase. For GIO, the beta gamma also subunits also have effects. So they go and they will close voltage-gated calcium channels, and they will go open potassium channels called GERK. GERK stands for G protein coupled, that's the G, inwardly rectifying potassium channels. So they're opening up a potassium channel. So overall, the effect of this is going to be hyperpolarizing. So remember we talked about the GABA B receptor opening up potassium channels. The GABA B receptor is metabotropic, but it is a GIO coupled receptor. So that's why it opens a GERK. That's how it produces a potassium current. So let's think about this more by thinking about morphine. Okay, so morphine signals through a receptor called the mu-opioid receptor. That's how the mu-opioid receptor is the main one involved in the pain relieving effects of opiate drugs like morphine. So morphine stimulates the mu-opioid receptor for endorphins. This receptor couples to GI. What that means is that this receptor, its G protein is GI. And so there's something called pertussis toxin. Pertussis toxin prevents GI activation. How would that affect how morphine changes membrane potential? Go ahead and think about that. All right, so let's think about what morphine would do here. So morphine is acting like an endorphin. When morphine is here, it would activate the alpha subunit of the G protein, and that would cause the beta gamma to go off and activate things. So we're going to lower the alpha is going to lower adenylyl cyclase levels, but that won't change membrane potential all by itself. What's going to change the membrane potential is the beta gamma opening up a potassium channel and closing a calcium channel. And so this is going to be, both of those effects are hyperpolarizing. So morphine would cause hyperpolarization. If you have pertussis toxin, you are going to block that. So by pertussis toxin stops that from happening. Okay. So now we have GQ. So GQ is going to cause release of calcium from the ER. How does it do that? So GQ activates the alpha subunit goes and activates something called phospholipase C. Phospholipase C acts on PIP2 in the membrane, and it um, creates diacylglycerol and IP3. The diacylglycerol can directly activate protein kinase C. The IP3 goes and it binds to IP3 receptors on the endoplasmic reticulum. When it does that, it opens this calcium channel, and the calcium comes out so that you have calcium release. And so we'll talk about calcium in the next video, but basically between these two, you increase protein phosphorylation, and now you have a bunch of this calcium inside the cell, and you're going to be activating a bunch of calcium activated kinases as well. Okay, so question for you here. You have recently identified a novel type of GPCR in neurons. You have in the cell, you fill the cell with a fluorescent calcium sensor. Remember, we talked about those in a previous video, and then apply this ligand, the ligand to this GPCR. So you want to see, does applying this ligand cause there to be more calcium inside the cell? If the ligand causes an increase in intracellular calcium concentration, the fluorescent calcium sensor would be green. So which G alpha subunit would be most likely to cause those increases in green fluorescence? Yeah, it is going to be GQ. So if this GPCR is GQ coupled, the alpha subunit would go and do all the things in the previous slide and cause release of calcium from internal stores. And so there would be green fluorescence as there would be more calcium inside the cell. All right, so I know that's a lot, but just kind of remember that like all those different receptors that we talked about, it can be boiled down into these three, and two of them act on the same system. They act on cyclic AMP. GS raises levels of cyclic AMP. GI lowers levels of cyclic AMP and also opens potassium channels. BQ, on the other hand, causes release of calcium inside the cell. All right, I hope that works for you.